So I love the UCSC browser. You can't not love the UCSC browser if you're in genomics, because it's kind of like not loving the air. You depend on it sort of too much. Anyway, but there are things that you can't browse with the UCSC browser, and so I'm going to just show you things which I contend are interesting and which you cannot browse using the UCSC browser, and how potentially different ways of visualizing data kind of come to the forefront when you realize this. So this is sort of the problem that motivated at least part of this work, which is, as Brad just told you, the genome is 2 meters long if you stretch it out. Sort of UCSC browser that was approximately, real scale, 1-to-1, would be about 2 meters long. That's a pretty large screen, but not inconceivable. Of course, you can't see anything on it, but still, there you have it. Of course, this thing actually is folding up into a 6-micron wide nucleus. So clearly, things are folding up in 3 dimensions. There's all kinds of long-range interactions that must be happening, et cetera, et cetera. Okay, so here's a picture also that Brad just showed you. This sort of tells us how, the textbook tells you that things fold up at large scales. So first you get the double helix, and then things are winding around chromatin, and then there's this thing called the 30 nanometer fiber, which people sort of understand rather less, and then there's this thing called the extended scaffold associated form, which people understand rather less, and then the condensed scaffold associated form, I guess that's the condensed extended scaffold associated form. And then finally you end up in the metaphase chromosome, and you realize that you're not actually, we've left the nucleus, Toto. We're not in Kansas anymore. Well, that's too bad, because actually it's quite interesting to figure out, because the problem that we had was we were trying to figure out how the genome folded up inside the nucleus. So this is obviously not answering the problem. So let's try to improve this little picture. So the first thing is there's this notion, which is kind of similar to the notion of a metaphase chromosome, called the chromosome territory, which is sort of the very, very larger scale structure of the chromosome inside the nucleus. So that's good. So this is an improvement. We're improving this visualization. Let's improve it further. Here's an improvement. Actually, we don't really know what's going on here at all. So this is an important sort of thing to realize, which is that you can make gorgeous visualizations. I mean, really remarkably beautiful visualizations. And they don't necessarily reflect the fact that here, the scientific community as a whole really, really, really knows what they're talking about. And kind of around here, we sort of don't. It would be interesting if you sort of colored in all visualizations with, percentage was somehow the color represented the certainty that this is actually real. And now let's think about this gap, right? So we know that the genome at the scale of about 10 base pairs looks like a double helix, and at the scale of about 100 base pairs, it's winding around chromatin proteins. And then we know that at the scale of whole chromosomes, like 100 megabases, it looks like a chromosome territory, which I'll tell you about. And then there's 5 orders of magnitude in between that we just don't understand. So that's a lot of room. So I'm skipping the section on prior art. There's been a whole bunch of work in this area, fantastic work on nuclear ligation assay and chromosome conformation capture. I just don't have time to get into it because I really want to spend as much time as possible talking about interesting ways of visualizing data and kind of zoom you through there. So okay, here's the problem that we're going to try to solve. We're going to try to figure out how things are folded up inside the nucleus, and we're going to do that by virtue of trying to figure out whenever 2 pieces of DNA, say this blue piece and this red piece, are nearby in 3D, we're going to try to figure out that that is the case and just read that out. So how do we do that? So first we start by cross-linking DNA, fixes sort of everything. It's kind of like Spider-Man shooting everything with a web shooter. Now you can't move. Then you cut everything with restriction enzymes. So now the genome's cut into 1 million little pieces, some of which are stuck together with the Spider-Man formaldehyde webbing. And now you do the trick, actually, in this protocol, which is when you cut with the restriction enzyme, you left sticky ends. So now you're going to fill in those ends with nucleotides, except one of the nucleotides is biotinylated. Great. So now you ligate, and now what you have is these circles. But the neat thing about these circles is, first, they're chimerical. Nothing like this exists in the genome. The red piece and the blue piece are physically proximate, but they're not sequentially adjacent. Now they're sequentially adjacent. Moreover, the exact point at which you go from the sort of blue to the red, you've got this conveniently located biotin. So what you can do is you can rip this thing to shreds and pull down just the biotins. And now you've got a whole bunch of junctions. And of course, you can sequence these junctions. Now, a single sequence, what does a single sequence tell you? It tells you that I had 20 million cells. And somewhere in the 20 million cells, this red piece of DNA and this blue piece of DNA were in physical contact. Well, that's not a lot of information. Of course, the great thing about sequencing is that you can do this many, many, many times. And once you've done this 50 million times, all of a sudden you actually have a fair bit of statistical information. So now let me show you the first thing that you cannot do with the UCSC browser, which is the natural data type here is just, it's not a one-dimensional data type. So what I'm interested in is, here's chromosome 8, right? We generate this for the whole genome, but here's chromosome 8. And what I'm interested in is how often does a given bit of chromosome 8 interact with another bit of chromosome 8. So here, for instance, each pixel represents a region of 1 megabase of chromosome 8 and all of its interactions with another megabase of chromosome 8. And its intensity tells me how many reads I saw. So for instance, if I'm starting out, I might say, OK, I'm interested in locus A. How often does locus A interact with itself? A great deal. That makes sense, because locus A should be close to itself in the sense that I'm close to myself and you're probably close to yourself. Of course, locus A is rather less close to locus B, still less close to locus C. There's some complexity here, right? There's polka dots, there's a little starburst. In fact, this sort of localization effect, right, that locus A is always close to itself, that holds for all loci on all chromosomes of all organisms. Whenever you've done a successful IC experiment, you should always be seeing these diagonals. But the critical point is this is really a fundamentally 2D data type. It doesn't like to live in the UCSC browser, and it points towards the fact that we might need new ways of thinking about proximity data. OK, so now I'm going to use this data to take you on a tour of the nucleus, if you will, as reconstructed using Hi-C. And I'm going to try to highlight at various points on this tour different sorts of visual problems that arise and the ways in which thinking about new types of visualizations helps us solve those problems. So we're going to not tackle all 5 of those orders of magnitude, but we're going to start to probe at that sort of largest order of magnitude, the chromosome territories. This is something that was known. And then we're going to kind of zoom in to the 10 megabase order of magnitude, the 1 megabase order of magnitude, and talk about some of the structural features that Hi-C suggests. OK, so let's start at the nuclear scale. And this is just kind of control to tell you that things are normal. Basically, when a chromosome is kind of wiggling around in the nucleus, it doesn't wiggle around throughout the entire nucleus. It kind of keeps to itself. Each chromosome keeps to itself in a little wedge. They look kind of like Trivial Pursuit wedges. And in fact, you can kind of see that. If you look at contact probability, as long as you're on the same chromosome, your contact probability is higher than if you're on different chromosomes, even if you're like 200 megabases away, right? There's always this gap. So that's actually consistent with this notion of a chromosome territory, which this is kind of the green guys there are the chromosome territories, right? They look sort of like Trivial Pursuit wedges if you play a fuzzy version of Trivial Pursuit. And you can kind of figure out roughly where these things are organized. And in principle, actually, you could probably take this map and try to make something that looks more like a microscopy image of this type. Although, actually, I kind of contend that for this type of data, it's just easier to read it off this sort of plot than to actually reconstruct something really fancy. But I think that's just because the data is at an early stage. OK, so let's zoom in now, take a look at the chromosome scale, see some of the features that we find. And so the standard way of thinking about this problem is that if you've got loci that are nearby, let's say 2 yellow guys, and they should be interacting a lot. They should be making lots of contacts. But actually, one natural sort of thing from the analytics standpoint is, well, let's suppose that you live in the same building as someone else. Maybe you don't shake hands a lot, but maybe you share neighbors. So there's a lot of people who you both shake hands with as you pass by, or who knows what, high five, depending on your age. I don't know. So suppose there actually might be a whole club of yellow guys, and a club of blue guys, and a club of red guys, a club of purple guys. They all share interaction patterns. By virtue of the fact that they share interaction patterns, you can figure out that they probably live nearby in the genome. So let me just kind of show you in practice what this does. So here's sort of a raw interaction map. Here's sort of, you include this big diagonal effect. So you kind of subtract that out, and you've got an observed over expected map. And now you do this sort of correlation trick for interaction maps, and you can actually see that this is dramatically improving the quality of the resulting data, right? I mean, all of a sudden, all of these little dividers are very, very, very clear, where you might have not had data at all. Now you've got data. And in fact, the vast majority of the points here are actually independently statistically significant. And what's important about this is that when you start to look at these proximity problems where the fundamental data type is genome-wide, it's genome-wide by genome-wide, so you're talking about the matrix that underlies all this is 3 billion by 3 billion. So I believe the total number of possible interactions is about 10 sextillion. 10 sextillion turns out to, amazingly enough, still be a larger number than the number of reads that a high-seq produces in a week. It's hard to imagine that those sorts of numbers exist, but in fact, it is bigger, and so it highlights the fact that whereas everywhere else in sequencing land, in some sense, you can kill the problem data-wise. You can just put as much data as you need and just kill the problem if you want. You can never kill the problem with these things. I mean, I don't know if this is third-generation sequencing. I don't know. It's going to be many more generations of sequencing before you can kill this kind of problem data-wise. There's some features. You can see these sort of opposing patterns. And as I said, the patterns suggest spatial neighborhoods. And so the fact that you've got two opposing patterns, and you see that genome-wide, suggests that chromosomes are actually segregated into two compartments. That's cool. And here, what's interesting is this is, I think, the one point in the talk where I can connect back to stuff that you can do one-dimensionally. Because the great thing is you've got these patterns, and it turns out by convenient and fairly straightforward fact of linear algebra that the principal eigenvector of one of these heat maps turns out to be a one-dimensional representation of which things have pattern A versus which things have pattern B. And so this is actually something that I can plug back into my UCSC genome browser, and I can see things like, oh, it turns out that if you've got one pattern, you tend to be accessible chromatin. You tend to be where all the genes are. So it turns out that these compartments are not just compartment A and compartment B. They seem to correspond to active and inactive regions. Cool. So now let's zoom in again. Let's look at what the data suggests us about the genome at the megabase scale. So let me tell you the following sort of fact. If life throws you a curveball, it will be no consolation that every curveball has a contact probability function. Still, it's true. In particular, let me tell you about that. So here we have a curve. It's a 1D thing that's folded up in higher dimensions. And you can see that sometimes it touches, kind of makes these long-distance contacts at different distances. This is a distance 3, 13, 213. And here, because it's colored from blue to red, you can kind of see how far apart you are in 1D by looking at how different the colors are. So now you can play the following game. You can say, OK, well, what is the likelihood that two things are touching if their one-dimensional distance is 1? Well, they're always touching because the curve is continuous. What about if it's 2? What about if it's 3 or 4 or 5? And you can actually make a little plot. And this plot is called the contact probability plot. And this is actually a pretty distinguishing characteristic of a three-dimensional curve. So we can do this for the human genome. We can plot contact probability, the likelihood that two pieces of the genome are touching, as a function of how far apart the two pieces of the genome are. And it turns out that this scales with, in a very sort of characteristic way, with approximately -1. And that's a diagnostic, right? So the fact that this is a line is interesting. The slope of the line is potentially a diagnostic for what the underlying fold is. So let me tell you how you can actually use this. So again, the unfolded polymer is going to range from blue to red. Now, how should the genome fold up? According to polymer physics, this is how polymers condense. They fold into this structure called the equilibrium globule. You can see there's colors all over the place. There's some green at this end. There's some green at the other end. Big, messy structure. Is this well organized? Is this how you would want to organize your data? Well, how should you organize your data? Let's suppose you're a library. You want to organize your data. You want it to be compact. You want all the books to be in one place. You want it to be organized. You want to be able to find the books when you want them. And you want it to be accessible. When you finally find your data, you should be able to retrieve that file. It shouldn't not be somehow accessible to you. You should be able to pull that book off the shelf and read through it. OK. Well, is this a good file system for your cell? Well, yeah, it's compact, but no, it's a disorganized mess. It's actually totally loaded with knots. All of this is forgivable if physics only gives you one option, but what's not forgivable is that this would produce a linear trajectory, exactly like the one that I showed you on the previous slide, but with the wrong slope. That was very depressing. A very therapeutic paper was published in 1988 by Grossberg and others. They said, hey, actually, we think a very, very long polymer might not fold into an equilibrium globule immediately. That's good. And they said, we think in the meantime, it's going to fold up into this structure. Of course, everyone agrees about the equilibrium globule, right? Because it's already been part of what got Paul Floria's Nobel Prize. So you're not going to disagree with that. But they were saying is initially, as a polymer is kind of getting to become an equilibrium globule, it's initially going to be a fractal globule. Now let's look at that. That's super dense, but yeah, it's really organized, right? Those beautiful monochromatic blocks. It's enormously accessible, actually. In theory, it should have no knots. And the slope, so I didn't bother to calculate the slope, but I was able to do a simple back of the envelope, and it turned out to be -1. So that's good. Now, before I move on in the talk, I'm using this word which, I don't know, it's kind of unnecessarily, I mean, maybe I should have it would be better if they'd called it like one of these Web 2.0 names that's like unintimidating, like Gidget or something like that. But they called it fractal globule, which is kind of not the best name. So let me sum up the physics for you in a very intuitive way. So I've told you, you take the fractal globule and you allow it to equilibrate, which in statistical mechanics happens using thermal energy, and it becomes this structure called the equilibrium globule. Of course, I don't know. That seems very foreign, but I actually claim that this is something that's completely intuitive because many of you went to graduate school, and in graduate school you probably ate the following because you have no money. So you eat this. Now, this is actually topologically very similar to the fractal globule, right? And so these guys who make the ramen noodles, they have a very similar problem to the human genome. So they've got all these noodles. They want to pack lots and lots of noodles into a 4-inch by 4-inch package. Does anyone have a sense of how much noodle, what's the total contour length of all the noodles in a packet of ramen noodles? 4-inch by 4-inch, 25 cents at CVS. Any estimates?
2 meters.
2 meters, okay. Does anyone think it's higher? 50 meters. Does anyone think it's higher than that? 50 meters is about right. It's about 170 feet, as was very laboriously demonstrated by a bunch of folks who actually boiled it, laid it end to end in a very large parking lot, and took very, very detailed photographic images, which they then posted on their blog, which is, of course, the point of the internet. So anyway, the point is, you have this structure, and I want to point out some topological properties of this structure. So you take this structure and you put it in water, because that's what you do with it, and then you're very, very, very hungry. So after approximately 10 seconds, you hope that it has started to cook. So you stick a fork in it, and you kind of pull back at some of the noodles, and you actually realize that nothing has happened because it's only been 10 seconds. It's only like in your stomach that it seemed like it had been a half hour. So you just kind of you release the fork and you put those noodles right back in, and it's like nothing happened. Because it's a totally unentangled structure. So you can remove little modules in a very simple way and put them back. Of course, if you're like me, you like to equilibrate this structure using thermal energy. Of course, I prefer to do it in a pot. And then you end up with this new structure at the very end. And I want to point out this is a topologically different structure, right? This thing was dense but unentangled. This thing is dense but highly entangled. And you can do a very simple experiment, which is just to stick a fork in it. It's delicious, but it's highly entangled. Anyway, so this also, I want to point out this is actually a visualization problem that I think has actually been solved, but maybe without folks in the audience actually noticing, right? Which is, I've been talking about this fractal globule, and I keep showing you pictures that look like this picture, but in fact, we didn't actually have that picture. And in fact, there were some nice related mathematical functions like this Hilbert curve, but we didn't have that picture either. Actually, the state of the art in drawing fractal globules at the time that we kind of made this discovery was this from the original paper by Grossberg and others. And I mean, it's wonderful, right? But I mean, really, nobody's going to get really excited about this particular picture. So we said we need to try to do better. And so I approached Leonid Murney and Maxim Akayev in the HST program here. Said, why don't we work together to try to simulate a polymer as it condenses? And then we worked with Najib Tarazi to make some images. Najib is over at Pixar by day, and he's the Clint Eastwood of polymer physics by night. Now you can see this polymer has folded up, and it's producing these nice monochromatic blocks. In fact, it spontaneously folds up into a fractal globule that has all of the properties which we theorized about, but now we have prettier pictures. Now, moreover, it's got, again, it's a modular structure, right? You can pull out you don't have to unravel the whole ramen noodle thing in the pot. You can just pull out a couple of noodles in your little experiment, and that's actually useful. Suppose you want to just activate one gene in this big, megabase structure. So here what we're going to do is we're going to attenuate the potential, the force of attraction that's keeping that light blue region together, and you can see that just that region starts to come apart. That's pretty cool. The rest of this thing is intact, and now in a moment, Najib's dynamic move is going to happen. And what's happening here is you can see that that cyan region is entirely unraveled. Now you're staring down the barrel of a fractal globule, which is largely intact. So you can really get a sense of the modularity of these structures. And so I sort of, I want to point out that, excuse me. So I want to point out that there's all kinds of visualization issues that arise in creating this visual. And I mean, Najib actually worked quite hard to make polymers that looked reasonably and moved reasonably and gave you some sense of intuition. And these are visualization problems that I think are, to some extent, new to DNA folks. Protein folks have been dealing with them, but they're new to DNA folks. And they require us to maybe go beyond their comfort zone. And there's multiple ways of trying to approach them. So for instance, for these structures, a natural thing to do is to try to print them out in 3D. And so here's fractal and equilibrium globules printed out in 3D, and you can look at them and get some intuition. It turns out that what's much better is we can actually print something out in a flexible material. So this is a 3D printout. It's not a fractal globule. It's actually a 3D Hilbert curve, which is a related mathematical structure. You can see it's very, very dense, but we printed it in a flexible material, so the whole thing comes apart entirely. Right? So this kind of thing, right, this sort of visualization, I mean, this is a visualization too. And these are actually really valuable for getting some sense of insight about structure and its dynamic properties. So we have to start thinking about, I think, some of these issues together. So let me sum up. There's this big void in our understanding of structure. We're trying to fill in that void. I think Hi-C is useful for doing that. And I wanted to just thank for this part of this, especially my close collaborator, Minka van Berkom, who got this technique working with me, as well as Louise Williams, who did a bunch of experimental work, and then Job, and especially Eric, for really encouraging me in this weird direction. But now I actually want to do something totally different. If I have a couple of minutes left, I want to do something completely different, because I've got a whole bunch of data visualizers trapped, which is great. So here's more stuff that you can't do at the UCSC browser. Now, I would venture to say that at this point, we've had a turn in this presentation, because I think the previous stuff you could imagine wanting to be able to do in the UCSC browser. And I would venture to say that the next stuff I'm going to show you, you don't even want to do on the UCSC browser, but it's still, you probably want to do it, just maybe somewhere else. This is joint work with Jean-Baptiste Michel. Again, big data, visualizing big data, and I think you can learn something about that potentially. I've learned something about it from trying to do this. So here are some things you can do at the library. You can read a few books very, very carefully. That's what most of us do. Here's another idea. Maybe you could just read all the books. That takes too long. So maybe we'll just read them very, very not carefully. I think that would make up for it. OK, so here's how we're going to do that. So all these authors, they've been writing for hundreds of years. They've published 129 million books. Google has been scanning all of these books. And so now they have them in digital format. And so what we did, of course, it would be very nice if Google could just release the full text of all these books. But see all those names like R. North and M. Twain and A. Kessel. I mean, in Google's brain, they're not authors, they're plaintiffs. So you can't actually release the full text of all these books. They said, OK, let's try doing something else. What we're going to do is we're going to track the frequency of words and phrases over time in these books. And this took us a whole bunch of years to get to work, but then it worked, and then they let us release the data, and it was good. And so let me show you why this data is useful. So this is the state of the art right now, or this was the state of the art like 6 months ago, which is, let's say I was interested in the verb thrive. And now thrive can be conjugated in two ways if I did it yesterday. I either throve or I thrived. Great. Now, if I ask this distinguished linguist, I say, hey, do I thrived or do I throve? He would say, oh, mostly you thrived, but some people say you throve. If I ask this other distinguished gentleman, he would say, oh, most people throve, but some people thrived. So I know that's roughly the state of the art in terms of public knowledge up to 3 months ago today. Well, but now we have this data. So now what I'm going to show you is actually just data, just raw data, year by year, single year resolution. And you can see this transition. This is called regularization in linguistics. And you could kind of tell that something like this had to be going on, but people could really see it. And now we can just see this stuff. I'm just going to throw this. I mean, this is a sledgehammer. So there's about 2 billion different trajectories like this that we tracked. So I've just shown you 2 of them. So the moral is that the actual total data set is 1 billion times cooler than this slide. So even if you're not impressed by this slide, right, still 1 billion times not impressive has got to be something. So anyway, so let me just show you a couple of things you can point this sledgehammer at. Let me tell you the history of 1950. Nobody was interested in 1950 in the 19th century. It was remarkable, actually, the fact that people just didn't care about 1950. In fact, until throughout the 30s, basically, no one really cared about 1950. And then in the 40s and late 40s, there was a buzz. People began to realize that 1950 was going to happen, and it was going to be big. So that was it. But then nothing really affected the history of 1950 like 1950. During 1950, it was pretty much the only thing people could think about. Everything they wanted to do was happening in 1950. All the stuff that they wanted to discuss was during 1950. And actually, for a couple of years after that, they kept talking about it. They were like, wow, you know, let's debrief, because 1950 was really exciting. Let's talk about all the things that happened in 1950. And then, kind of towards the mid-50s, 1950 just got a little bit passé. And it actually got passé quite rapidly. And we can measure how rapidly it got passé by just measuring the half-life of this curve. And in fact, we can measure the rate at which all years get passé. They all look more or less like this. We can measure the rate at which all years get passé, and they seem to be getting passé faster, out with the old. Other things we can do is another totally different example. So Marc Chagall, Jewish artist, this is what a person looks like usually when they're becoming, like a famous person, like Brad Bernstein, who just spoke. So this is what you look like when you're becoming famous. And the following trajectory is something you'd never, ever, ever see. This is Marc Chagall in German. And you can see, I mean, obviously 33 to 45 is the Nazi regime, and you can really see it basically as a nadir during that period. You never see this. I mean, this is really political suppression in action. It's not just for distant history. You can see this for Tiananmen Square as well. There were 2 events in Tiananmen Square, the 76 event, the 89 event. In Chinese, you can see a big signal around the 76 event where people started talking about it. In English, you see a little signal for the 76 event. You actually see a huge signal for the 89 event, the massacre that we talk about. You don't see that at all in Chinese. And so what you're seeing here is not a single anecdotal piece of evidence, but actually statistical analysis of a huge portion of all Chinese books ever published that's telling you that this is happening. And now I actually want to sort of turn and talk more about what visualization can do for you, leveraged in the right sort of way. So we published this paper. That was cool. But what we did that I think was really good is about like 10 days before, we took our prototype browser and we managed to convince Google that they just had to make this available online. And so we created this very, very, very simple interface. You can just type in whatever word or phrase you're interested in, and it will tell you how frequently it's been used over time, like when did people start referring to the United States in the singular as opposed to the plural, which people have debated for a while, but now you can just check. You can link directly to the original primary sources, so you can do primary source stuff. This is the best query I've ever seen. People in the 1800s, people in the 1700s, they were constantly trying to put their best foot forward, to do their best, to strive for their best. And that really stopped right around 1800. Now, it's not because people were striving for mediocrity, but instead they started to put their best foot forward. Now, what's going on there? We actually explained what was going on there in our 100-page tome about this. But I think actually a testament to the fact that this visualization is nice and simple is the fact that no one actually read anything that we wrote. They just played around with this thing and discovered this best thing and realized for themselves actually that this is it's a fact about typography. So the English S used to be written, when it appeared in the middle of a word, it used to be written like an integral sign and gets digitized as the letter F. And so this actually very clearly is telling you when typography switched over in English. And it actually switched over at clearly different points in various different languages. People have been using this like crazy. So here's a marketer. He said, look, we need to think outside the box. We need to incentivize and strategize so we can synergize and touch base in a goal-oriented way about all the different things that the Ngrams viewer can do for us. There's also more serious stuff that's been done with this. So the folks at Wikipedia, they said, look, let's look at our articles. Let's look at how good our articles are as a function of how famous people are in the Ngrams corpus. And what they found is they do a good job, better articles for more famous people. But they said if we take comparably famous people who are male versus female, we actually are writing way the heck better articles about the men than about the women. Start, stub means very bad article. Letters means pretty good. So that's very striking. So this was really the first time that they could quantify the extent of the bias in Wikipedia. And actually, I mean, this visualization, I just want to kind of make this point. So this visualization has actually become so I don't know, people like it so much that, like, I don't know, so this guy's like an editor, senior editor at The Atlantic, and he just wrote this about, I mean, there's obviously a nuclear crisis going on, and I mean, basically this article is just, he ran a bunch of n-grams. And people have been using the n-grams thing a lot, but I think what's neat is that, I think we kind of hit, partly by, dumb luck, but we hit on something where the visualization in and of itself is so compelling that people don't even write text anymore about these. They're like, just look at this data. It's a good thing. And so, I mean, I think for that reason, I want to kind of highlight the following n-gram. So here's megabyte, gigabyte, terabyte, petabyte. Data is getting bigger and bigger. But what you can see is really that as that is happening, it's becoming more and more and more important. We can't read it all. We need to come up with clever ways of visualizing all the data so we can deal with it. With that said, yeah, we've got to do everything else, but I just want to thank all the people who are involved in this research, especially my collaborator, Jean-Baptiste Michel, Yuan Chen, who did a Google internship working on this stuff, John Orwant, Dan Clancy, and Peter Norvigel at Google, who agreed that this was not stupid. That's all I had to say.